Hello, everyone. Thank you for attending today's webinar, Advances in Precision Medicine in Lupus and Rheumatoid Arthritis, presented by Sengenics. I'm Ross Yule, and I'll be moderating today's webinar. Our speaker today is Dr. Myles Lewis, PhD, FRCP, Reader in Rheumatology at Queen Mary, University of London. You can read their full bios on the left side of your window by selecting the Speakers tab. Just a few technical notes before we begin. The webcast is being streamed through your computer, so there is no dial-in number. For the best audio quality, please make sure your volume is up. This webinar is being recorded and will be available on demand within 24 hours after the event. Time permitting, we will follow the presentations with a Q&A session. Please submit your questions using the Questions and Answers tab on the left side of your screen. Okay, now let's begin. Dr. Lewis, please go ahead.
Thank you, Yul. So my work is both on lupus, but also on rheumatoid arthritis. So I'm going to be presenting data from a number of different papers over the last couple of years from my own group, but also our extended group at Queen Mary, which is headed by Professor Pitsalis on rheumatoid arthritis. This initial work in lupus was also done in conjunction with King's College London. So lupus is a severe multi-system autoimmune disease, and although it's very well known to affect the skin and joints, and the slide shows some of the typical disfiguring skin lesions and joint problems that can occur, it also can affect multiple different internal organs such as kidney, lungs, heart, brain, and can also affect the neonate. It's a surprisingly common condition. It's much more common in women, and it also has a very strong predilection for different ethnic groups, particularly non-Europeans, such as either Asians or African ethnicity groups. And one of the big problems in lupus is that it's treated with potent immunosuppressive drugs, and at the moment, it's very difficult to predict whether a patient is going to respond to a particular drug, and this is something that is of major interest in the field, is trying to predict who is going to respond to which drug. So in clinical use, there are a number of autoantibodies that are widely used for both diagnosis but also prognosis in lupus. The most well-known of these include double-stranded DNA, which is associated particularly with nephritis, but there are other antibodies that are commonly used as part of the diagnostic workup, including Rho and LAR, as well as Smith and RMP. Now, Rho and LAR are not specific for lupus. They can also be seen in Sjogren's, and to some extent are seen in other autoimmune diseases such as rheumatoid. However, they are quite common in lupus and therefore quite useful in terms of clinical practice. And then the other antibody that is commonly performed are cardiolipin antibodies, which have their own set of associations, including the antiphospholipid syndrome. So this work that was performed in conjunction with Professor Tim Weiss at King's and Oxford Gene Technology, which was a company that had developed under the guidance of Jonathan Blackburn an advanced microarray for detecting autoantigens. Now, this autoantigen array contains over 1,600 full-length human proteins, and these proteins have been expressed in insect cell lines. And this is particularly important as previously has generated protein microarrays of older generations used proteins that were generated either in E. coli or yeast, and these proteins often do not fold correctly, and previous studies really did not find very many autoantibodies through this method. Another key point about this technology is that the proteins are expressed with a BCCP folding tag. Now, this type of tag is particularly important because it ensures that only correctly folded proteins are actually linked to the microarray. And this enhances the ability of the microarray to detect correctly folded proteins as antigenic. So I'm just going to describe the study that was performed in lupus. So the initial American cohort contained 90 control patients and 85 lupus patients. And then the UK cohorts contained approximately 200 lupus patients and approximately 200 controls. And there was also a confounding disease group, which included a number of different patients, largely with rheumatoid arthritis, but also with other connective tissue diseases. And the purpose of that was to determine specificity of some of the autoantibodies. So this is just a flow diagram demonstrating how the cohorts were organized. The cohorts are obviously grouped into control and lupus, but then when we actually went on to perform more detailed data analysis, it became clear to us that because ethnicity was a confounding factor, we needed to actually partition the data 2-to-1 into discovery and validation with merging of the cohorts in order that we could have equal numbers of the different ethnic groups across each cohort. And so this is the initial primary data to come out of the analysis. On the left, you can see a volcano plot showing in blue a significant number of proteins that were recognized by the microarray as autoantibody targets. And this is using an FDR cutoff of 0.05, so adjusting for the number of antigens on the array. And I think the first thing you can see is in the top right, you can see TROV2, which is Rho, and SSB, which is LAR. And so the autoantibody array is able to validate very commonly known autoantibodies such as Rho and LAR, and also other autoantibodies that have previously been described, including PSME3 and various other autoantibodies that are also known in the literature. And then on the right, we're demonstrating in the validation cohort those proteins that were identified, and the ones that are marked in red were validated in both cohorts. And this is just some individual results for Rho and LAR, and also some other autoantibodies that have previously been identified, showing that the array is able to replicate these results both in the discovery cohort, but also in the validation cohort. And in this plot, we're demonstrating antibody positivity using the control cohort as the cutoff. And again, on the left, you've got the discovery cohort with the autoantibodies ranked in terms of those with the highest positivity down to some with the lowest levels of positivity. And again, in the middle panel in red, you can see how positive these were within the validation cohort. And then on the right, this is a demonstration of the number of positive autoantibodies per individual. And essentially, you can see that that some individuals have substantial numbers of autoantibodies, whereas as you go up to higher numbers of autoantibodies, fewer individuals show that level of percentage. Now, in this heat map, we were able to see a distinct clustering of the lupus patients by the autoantibodies. And this was some of the most interesting results because it suggests that there may be subgroups within the lupus patients that have not previously been identified. We've always suspected that these subgroups may exist, and speaking as a clinician, because we know that patients often have very different phenotypes in lupus. It's a very heterogeneous disease, and you have some patients in clinic who have only really got skin and joint disease, and other patients with very severe organ involvement, such as kidney involvement. And it can be quite difficult to understand why these these marked levels of heterogeneity occur within clinic. And I think what we're seeing here is perhaps some of the underlying mechanism in terms of the heterogeneity. And so what we found was that there were 4 separate groups which can be clearly seen by clustering. Group 1A, which consisted of a group of patients who were strongly Rho and LAR positive. Group 1B, which consisted of patients who had other autoantibodies such as Smith and PABPC1, which has also previously been identified as a lupus autoantigen. But what was really striking was that we saw these 2 other groups, SLE group 2 and SLE group 3, and largely these contain novel autoantigens that have not been previously described. In this slide, the cohorts have been divided into the discovery and validation, and you can see that SLE 1A, 1B, 2 and 3 are still visible within both the discovery and the validation cohorts. If you do a correlation of the autoantibodies, this demonstrates the underlying clustering, and you can very clearly see these 4 blocks of correlation showing that you have antibodies that cluster together in distinct groupings. We then went on to perform other methods of looking for structure within the data using principal component analysis, and again the groupings of patients into 4 subgroups were visible by PCA. We can see in red patients that fit with SLE 1A, in purple the SLE 1B patients, in green the SLE 2 patients, and in blue, going away from the screen, the SLE 3 patients. And this plot is available as a rotating plot that's animated if you read the actual original paper on the website of the Journal of Autoimmunity. Again, the loading scores for the principal component analysis also demonstrate that specific autoantibodies are associated with the different clusters. So, for example, you've got Rho and LAR strongly associated in the top right of the left-hand panel with the SLE 1A cluster, whilst you can clearly see autoantibodies associated with the different colors associated with each cluster. So the autoantibodies associated with cluster 1B include Smith, and these are in purple, whilst cluster 2 are in green and cluster 3 are in blue. And again, a three-dimensional view of this plot, which is shown on the right, very clearly shows how these autoantibodies are segregated. So we then went on to look at subphenotype now that we had identified these novel autoantibodies. And one of the interesting things that we could see is that there were differences between the patients in their level of ANA and DNA antibodies. And in particular, looking at the bottom left, you can see that the 1A group is strongly ANA positive, and similarly the group 2, which is in green, are also pretty strongly ANA positive. If you look at DNA positivity, that was strongest within SLE group 2 and least strong within SLE group 3. And we then went on to analyze this in a bit more detail. And one of the striking findings was that the large block of largely novel autoantigens in group 3 tended to be seen in ANA negative lupus as well as in DNA negative lupus. So this suggests that these autoantigens mark out new autoantigens for lupus patients, which may help with diagnosing the disease in patients who are either ANA or DNA negative. We then went to look to see if there were differences between the autoantibody clusters and subphenotype in terms of organ involvement. And we found some very interesting findings in terms of differences between arthritis, for example, which was higher in group 2. We also saw that pulmonary involvement was higher in group 1A. That's actually been reported before. we saw that renal involvement was highest in group 1B, and thrombocytopenia was highest in group 1A, whilst neurological involvement was lower in group 1A. And then in the top right, you can see that specific autoantibodies, and I've just shown a selection here, were associated with being able to detect differences in terms of the amount of patients who had different organ involvement. So, for example, patients who are PRKRA positive were more likely to have arthritis, whilst patients who were IGF-2BP3 positive tended to have more renal involvement. And this suggests that these autoantibodies may have an important role for helping to anticipate whether patients go on to get significant renal and organ involvement of different kinds. We also saw there were differences in terms of the positivity to different autoantibody clusters with ethnicity, as shown in the bottom right. And I'm just going to come on to some more analysis of that in the next slide. So when we compared the autoantibody positivity by ethnicity, we did see some striking differences. And we saw that a significant number of autoantibodies had higher levels of positivity in different ethnic groups. In the top left panel, you have a group of autoantibodies that were most strongly positive in Afro-Caribbean lupus patients. In the middle left panel, you have autoantibodies who were predominantly positive in Hispanic lupus patients, whilst in the bottom left cohort, you have a third group of autoantibodies that had highest positivity in European or Caucasian lupus. And the middle panel just shows some specific results that show distinct differences between the ethnic groups. On the right, we have a linear discriminant analysis, which is similar to a PCA analysis. And again, it's possible to differentiate that the groups of patients by ethnicity show clearly distinct patterns of autoantibody positivity, with Afro-Caribbean lupus patients with the red dots in the bottom left of the three-dimensional plot, whilst the Hispanic patients are in the top right, and the European lupus patients are in the top left, and the controls are in the bottom right. So we then went on to try to refine the autoantibody panel. And so this is a restricted set of autoantibodies. So it's not the whole panel of autoantibodies. It is restricted to those that were positive in both the validation and the discovery cohorts. And in this subspace plot, you can see the positivity amongst SLE1A, Group 1B, Group 2 and Group 3 very clearly. And this clearly shows the differences between some of the autoantibodies and how they're positive in different groups. However, when we compared this across ethnicity, you can see that there are similar differences. So AA, which represents African American or Afro-Caribbean, HA, which is Hispanic, and EA, which is European ancestry, clearly show different patterns of positivity for these autoantibodies. So then we used machine learning to develop an autoantibody panel. And this particular panel, which went through a number of different iterations, which began as a 56 antibody panel, and then after refinement ended up as a 26 antibody panel, showed significant improvements in area under the curve for ROC analysis when we were attempting to predict lupus patients from controls. And so the ROC curves in the top left show that using a mixture discriminant analysis model, you can see that this had the highest area under the curve in terms of prediction with an AUC of 0.86. And that's compared to an elastic net model, which was 0.773. And then if you compare that to the other autoantibodies that were used in terms of ANA and DNA antibodies, you can see that the MDA model has the highest level of prediction. And in particular, if we set a specificity of 90%, the MDA model had the highest sensitivity, with a sensitivity of 67% in comparison to the best sensitivity when using either ANA or DNA, which was 47%. And so this really highlights something that we find in clinical practice, which is that ANA is sensitive, but it is really not a very specific test. And this is a big problem that is very well recognized by rheumatologists. And so we believe that this reduced antibody panel with 26 antibodies shows good discrimination and could be used as a novel diagnostic. One of the interesting analyses that we then went on to do when we looked at the panel of autoantibodies that were positive from the array was that we did pathway analysis and protein-protein interaction analysis to try to understand if there were links in terms of the actual function of the autoantigens and if they were linked in any way functionally. And to our surprise, we found that cluster 2, that's it's a block of novel autoantibodies associated with DNA positivity and renal disease. And we saw that this group had a number of different proteins that were all linked together through a TGF-beta signaling. And this was a surprise to us, but was a really interesting finding, suggesting that this has a potential functional link to understanding the pathogenesis of lupus in these patients. And then we looked in cluster 3, which again is largely consisting of novel autoantigens. We saw that these were strongly associated with NF-kappa B and toll-like receptor signaling, as well as apoptosis and B and T lymphocyte differentiation. And so I suspect that some of these autoantigens are perhaps a sort of missing link, if you will. These are autoantigens that are associated with lupus as discovered by the new array and do clearly link to lupus pathogenesis, but perhaps because the positivity isn't as common as antigens such as Rho and LAR, they've not previously been described before. And this is a hierarchical clustering plot that shows how the autoantigens cluster, and it's just a way of showing some of the key autoantigens. So, for example, the ones that are known about, which are very widely used already, include the ones in red, Rho60, LAR, SMN1, which encodes for one of the proteins within the Smith complex, PSME3, which is part of the Chi antigen. But the novel autoantigens of interest include the purple ones, which are largely mRNA editing or transcription factors, the autoantigens in green, which include the TGF-beta or Wnt signaling associated autoantigens, and then these two blocks in blue, which are associated with toll-like receptor signaling, NF-kappa B activation, apoptosis regulation, or B and T cell development. So just to put this into the wider context, stratified medicine, there's been a lot of focus on this in the last few years. And just to explain what this is, stratified medicine is the concept of trying to define clinical and genetic disease subgroups within a disease such as lupus, and using different methods to do that, not just clinical information, but biomarkers such as either genetic or serum biomarkers, or even transcriptomic biomarkers, and performing in-depth omic analysis, either based on blood or tissue, and trying to segregate the patients into clusters and groups to understand how these patients respond to different drugs, and then to figure out whether, if you are able to define subgroups, whether these patients will actually respond differentially to treatment, and thereby identify more precisely the correct treatment for the correct patient, which is the actual aim. And in this context, an interesting paper was published by Bankero et al. In 2016 in Cell, performed extensive microarray analysis on blood samples from a relatively large lupus cohort. And perhaps one of the interesting things about this cohort was they had longitudinal data, so they're able to track the gene expression over time. And they used WGCNA, which is a system for grouping genes into modules so that you can see functionally how they behave, and they then performed extensive clustering. And the interesting thing that they found was that, perhaps similar to our study in terms of the autoantibody array, this time in terms of gene expression, they could see different clusters of patients with different blocks of genes associated, and they grouped them into myeloid lineage gene modules, interferon response gene modules, modules associated with plasmablasts and antibody secretion, lymphoid lineage modules, and erythropoiesis. And they were then able to cluster the patients. Now, in this clustering, they actually defined at least 7 different subgroups, but I think if you look carefully at the actual heat map in the center, you can see that there's quite a lot of overlap. So the more highly expressed modules, which are in yellow, compared to low expression of gene modules in blue, you can see that some of the subgroups show overlap between the different modules. So I think perhaps the difficulty here is that different subgroups can be overlapping, and therefore it can be difficult to define exactly how many subgroups there are. And this is something that I think is potentially work for the future, is that rather than defining subgroups of patients in whom maybe they have an increased interferon signature and an increased plasmablast signature, it might be better to think in terms of each of these modules more as an axis rather than necessarily an individual subgrouping. So just to summarize this section of the talk so far, we found that using an autoantibody array, we were able to identify novel antibodies which redefined lupus into 4 subgroups, and this matched 4 well-clustered groups of antigens. There is additional evidence from transcriptomics that there exist subgroups of lupus patients. We also showed that the antigens themselves clustered, and they demonstrated very clear functional protein-protein interactions and pathway associations. Now, 3 of the antigen clusters are largely novel. So although cluster 1A included old antigens that are very familiar to everybody, including Rowe, Lahr, and Smith, cluster 1B included antigens involved in RNA processing and DNA and chromatin interaction, whilst cluster 2 included a number of autoantigen proteins which are involved in the TGF-beta pathway and Wnt signaling, whilst cluster 3 included proteins involved in lymphocyte development, toll-like receptor signaling, and NF-kappa B activation. And 90% of ANA negative individuals who were diagnosed as having lupus fell into subgroups 1B and 3. And so this is a really important finding. It suggests that some of these novel autoantigens really will have importance in terms of identifying patients that are not captured by existing assays. And a novel biomarker panel of around about 20 to 25 autoantigens showed significantly increased diagnostic performance compared to ANA and or DNA antibodies. So I'm now going to come to talk about some of our work on rheumatoid arthritis, and this is work that's been performed in collaboration with Professor Costantino Pitsales, who heads up our department at Queen Mary in rheumatology. So why do we need to look at precision medicine in rheumatoid arthritis? Well, rheumatoid arthritis is less clinically heterogeneous in terms of organ involvement, but we do know that the disease is highly heterogeneous in terms of evolution and treatment response, and also in terms of pathology under the microscope. We also know that approximately 40% of patients do not respond to current medications. And the biggest problem at the moment is that we're unable to predict which patient is likely to respond to which drug. And so at the moment, patients typically start on methotrexate or other DMARDs. Approximately 40% or more of these patients will fail. And in the UK, these patients largely go on to anti-TNF. And again, another 40% will fail and then go on to other biologics. And subsequently, another 40 to 60% will fail and will either be defined as having refractory RA or will be put onto substandard treatments where the disease is insufficiently controlled. And the biggest problem at the moment is that this pathway is largely trial and error. It is not done in a way where we identify which patient actually needs which drug and start that drug straight away. So a number of years ago at Queen Mary, we developed a number of cohorts using funding from MRC, NIHR, and Arthritis Research UK, which is a charity now known as Versus Arthritis. And we set up three large cohorts in RA, an early arthritis cohort known as PEAK, or Pathobiology of Early Arthritis cohort, and an established RA cohort in patients who had failed DMARDs and then were randomized as part of a clinical trial to 3 drugs. And this cohort is called STRAP. The patients underwent a synovial biopsy and then were randomized to either rituximab, tocilizumab, or an anti-TNF. And then the final cohort is a cohort called ARFRA. And these are patients who have failed anti-TNF and were then randomized as part of an RCT into a trial in which they were given either rituximab or tocilizumab. And the really important point to note about all these cohorts is that all the patients had a synovial biopsy prior to the treatment that they were subsequently given. And so part of the rationale for looking at these results is to try to understand whether, on the one hand, there is some consistent explanation as to why some patients respond or don't respond, in which case there may just be some type of phenotype in which you have patients who have more severe disease who don't respond to drugs in general, or is it some type of disease endotype in which you have different patients who have different pathways that are active and they therefore need to be treated with different drugs and could respond to a different drug if they'd been given that from the outset. And that was really part of the rationale for developing these cohorts. Now, in terms of endotypes, we now think that the endotype system may be more complex than was previously thought. There may be drug-related endotypes. In other words, patients may have characteristics so that they may have higher levels of TNF signaling and therefore more likely to respond to anti-TNF, for example, or perhaps they have higher levels of other pathways being active, such as IL-23, IL-17 axis, or IL-6, and therefore actually respond to differentially to different drugs because of these different drug-related endotypes. Or there may be specific pathways that are active because of different pathogenic processes such as apoptosis or inflammation, and these may link to individual disease endotypes. And then finally, there may be molecular mechanisms. And of course, this then leads directly into differences in terms of either genetics or epigenetics between individuals that may explain why there may be underlying differences in either gene expression or protein levels between patients. And so we realized fairly quickly that in order to understand and dissect these cohorts properly was going to require a very big effort, not only to collect data as part of biomarker-driven randomized clinical control trials, but actually to do multiple levels of omic analysis, including gene expression, serology assessment, genetics, and so on. I'm just going to describe some of the key findings within the PEAK cohort, which is the cohort on the left. So the really important point to make about PEAK is that this cohort is a group of patients who are completely naive to treatment. So the biopsy was performed before these patients had steroids or any form of immunosuppressive drug. And this is a really important and unique cohort. There are other small studies that have looked at synovial biopsies from patients, but largely these other studies have been performed in patients with long-standing rheumatoid arthritis, and usually the tissue is taken from joint replacement tissue. PEAK was really the first study of its kind to look in early arthritis in treatment-naive patients. And initially, we analyzed this data using histology, and you can see in the top left that we found 3 different subgroups. A lymphoid group, which are patients who have B and T cells on their synovial tissue biopsy. But you can also see that these patients often had strong levels of CD68, which is a macrophage marker, as well as CD138, which is a plasma cell marker. Then the middle group, the myeloid group, which represented about 40% of patients, these were patients who had infiltration of macrophages, as shown by CD68 positivity, but they lacked B cells. And then finally, we identified a fibroid or portioimmune pathotype, and this is a group of patients who lacked immune cell infiltration, did not have B or T or macrophage infiltration. And so these patients are characterized by increased numbers of fibroblasts within the tissue. One of the striking things is that if you look at these patients just with the pure clinical markers, such as the number of swollen joints or tender joints, these patients are indistinguishable. So all of the patients had high disease activity as measured by their DAS28 score. The average DAS28 across all 3 of those subgroups was over 5. And yet these patients have very different pathology when you look under the microscope. So then we performed RNA sequencing of this cohort, and RNA sequencing was available in approximately 90 patients. And you can see in the heat map in the middle that there were distinct clusters associated with the different histology markers. So the histology tracks are shown in the top of the heat map, and you can see that they correlate with the gene expression, which is shown in the lower part of the heat map in these blocks. And so we were able to recapitulate the identification of these 3 subgroups within the RNA sequencing performed on the synovial tissue. And on the right is a principal component analysis showing that the fibroid group in green are on the right-hand side of the PCA plot, whilst the lymphoid group in blue are on the left-hand side of the PCA plot, whilst the myeloid group probably represents some type of intermediate group, and those red points are mostly in the middle, in between the blue and green groups. So we went on to develop an analytical platform so that this data can be shared with the community. So the link shown here is for a website. This website is public and live, and you can inspect the data from PEAK. And the website also provides useful links to a number of different publications to have come out of PEAK, and allows you to analyze the data and put in a gene of interest and see how that correlates with clinical parameters or histology outputs. And it's a very functional website. It allows you to ask a lot of questions about individual genes, and it's really been very useful both for ourselves in terms of understanding what individual genes are doing within synovium, but also correlating it with the clinical data. So just to summarize some of the key findings from this paper, which was published in 2019 in Cell Reports. So we did see that the RNA-seq showed good correlation with the histology markers. One of the first things that we set out to do was to try to understand how the RNA-seq was behaving at more than just a sort of individual gene level. So we used a very large public data set called Phantom5, and we developed our own way of deconvoluting the RNA sequencing to try to measure the expression of different cell types within the synovium through RNA-seq analysis. And so we identified modules that contained between 40 to 70 genes that were highly specific for each of these cell types, and then correlated those gene modules against histology. And so in the top, you can see on the x-axis, you have the histology markers, whilst on the y-axis, you have these gene modules that are specific for different cell types, such as B cells, plasma cells, T cells, macrophages, and mast cells. And so we were able to validate this approach, showing that the gene modules within the RNA sequencing showed good correlation with our histology markers. However, one of the big advantages of using the RNA sequencing is whereas the staining of the tissues is very time-consuming and it's very intensive, requiring a number of different technicians, with the RNA sequencing, we're able to look at a very large number of different cell types. So, for example, we are able to get information on regulatory T cells, synoviocytes, and other different cell types, such as endothelial cells, and see how these correlated with the clinical parameters. And what we found was some very interesting findings. So we could see that plasma cells, as measured by a plasma cell gene module, showed correlation with CCP titer. We saw that a monocyte module showed correlation with pain score as measured by the visual analog score, the VAS. We saw that plasmacyte or dendritic cells within the synovium were correlated with ESR, and the endothelial cells also showed correlation with ESR. And then one of the striking things about PEAK is that we had data in terms of ultrasound and X-ray, and we were able to see that some of these gene modules, such as the plasma cell module, showed strong correlation with ultrasound parameters, whilst the synoviocyte module was inversely correlated with ultrasound synovial thickness and inversely correlated with ultrasound power Doppler. This is very striking to see gene modules correlating with ultrasound parameters. And then in the bottom row, we can see that some of these gene modules also correlated with X-ray results at baseline as measured with the total Sharp score. So on the bottom left, you have the B cell module showing a correlation with X-ray damage at baseline. Now, one of the key things we wanted to understand was what is actually underlying the different pathotypes, these three different subgroups, the lymphoid, myeloid, and fibroid pathotype. And so we initially started doing standard differential expression in both synovium, in the volcano plots on the left, and then in blood, in the volcano plots on the right. However, we rapidly came to the conclusion this analysis was going to be very complicated because we had three different groups. And one of the key takeaway things to say about doing gene expression analysis and RNA sequencing is you have to be adaptable and be versatile and develop novel analytical tools, because these data sets are very big, and if you don't do that, you won't be able to get all the information out of them. So this is a novel type of analysis. It is a three-way volcano plot, and it shows this very striking finding. On the left, you can see the difference between the three pathotypes within the synovial gene expression. So the blue dots, each dot represents a gene, and those blue dots are genes that are upregulated within the lymphoid pathotype, whilst the yellow dots are genes that are upregulated in both the fibroid and the myeloid pathotype. The green dots represent genes that are specific to the fibroid pathotype, and the red dots represent genes that are specific to the myeloid pathotype. So instantly with this plot, you can see that there is some overlap between the myeloid pathotype. It has some overlap between fibroid and some overlap between lymphoid. In contrast, if you look at the RNA sequencing gene expression in blood, we saw very little difference in terms of difference between blood genes comparing the different pathotypes. However, even though there was such a small number of genes, those 8 genes are of great interest because they represent interferon response genes. So this suggests that an increased interferon response signature in blood is associated with the lymphoid pathotype. So we then did module analysis. So this is similar to the WGCNA analysis that was performed by Bankero et al., which I described earlier as part of one of the lupus studies. So on the left, you can see gene modules in the RNA sequencing in synovium. And this rather recapitulates what we'd seen in terms of the histology. In other words, B and T cell gene modules and associated gene modules such as CD28 co-stimulation being associated with a lymphoid pathotype, whilst we found Hox cluster, Wnt signaling, and various extracellular matrix-associated pathways associated with the fibroid pathotype. However, when you do gene module analysis, you do get greater power. And so this time when we looked in blood, we were able to see different blood modules associated with the different pathotypes. And perhaps the most striking of these were the top of the plot. On the right, you can see that there were TLR and inflammatory signaling within blood was associated with the myeloid pathotype, whilst in blue, the type 1 interferon response and antiviral response was associated with the lymphoid pathotype, whilst in green, we saw that a reduction in B cells was associated with the fibroid pathotype. And that's something that actually has been recapitulated in FACS. Of interest, we also observed that the RNA-seq expression was associated with different clinical outcomes. So we could see that there was a significant difference in terms of anti-CCP positivity, and we saw that plasma cell genes were upregulated in synovium in the CCP positive patients, whilst pro-plasma cell genes were also upregulated in those patients who went on to progress in terms of X-ray damage at 1 year. We then went on to see whether there was a difference in pathotype over time and how this related to response. And we found that although generally the pathotypes were stable, and you can see in this Sankey flowchart in the left, you can see that most of the patients stay within the same pathotype. However, we did notice that some patients would either increase their pathotype going from, say, myeloid to lymphoid, in other words, going to a more inflammatory pathotype, or occasionally decrease their pathotype going, for example, say, from lymphoid all the way down to the fibroid pathotype. And when we correlated this shift in the pathotype against the change in DAS28, we could see that there was a very clear difference. So those patients who tended to have a more inflammatory pathotype or the same pathotype as before, they responded less well over time to DMARDs, whereas those patients in whom the pathotype became less inflammatory showed a much greater response. And so the shift in pathotype was related to treatment response or failure. Now, a number of other papers have been published in PEAK, and for lack of time, I won't go through all of these, but I will make this important point that in these other papers, a number of which have been published in Annals of Rheumatic Diseases, we moved to measuring the synovial gene expression using nanostring panels. RNA sequencing is a relatively expensive technology, but it's also very difficult to standardize, so it could be very difficult to use in clinical practice, whereas nanostring panels have already entered clinical practice. For example, there is a breast cancer nanostring panel which can be used prognostically as well as in terms of helping to define correct treatment for breast cancer patients. And so our aim by moving to using nanostring was to see whether some of these panels could be used to predict disease severity. And that's exactly what we were able to show. We could show that a particular group of nanostring genes in combination with clinical parameters such as a rheumatoid factor and swollen joint count was able to predict X-ray progression at 12 months. And we also found that a nanostring panel was able to predict whether patients would need to go on to require first biologics. And so in both of these clearly delineate a group of patients who have more severe disease in the future, and yet this is all predicated on their baseline synovial biopsy, suggesting that if this was to be turned into a clinical assay, we would be able to identify these patients early and then put them onto the appropriate treatment early. So just summarize at this sort of stage, we found that there was distinct differences in RA gene expression that was linked to these histopathotypes, and they clearly existed within the RA synovium. We could see that these gene modules reflected the cellularity and inflammatory milieu, and the modules correlated with clinical activity and future clinical outcome. Specifically, we could see that a reduction in inflammatory pathotype could be associated with remission, whilst a worsening pathotype was associated with treatment failure. And we identified specific gene signatures to predict X-ray progression, DMARD failure, and first biologic use. So now I'm just briefly going to come on to some of the most recently published results, which is in the R4RA clinical trial. And we were very pleased that this was published earlier this year in The Lancet. So just to explain the primary aim of the trial, this is the first biomarker-driven trial in rheumatoid arthritis, which was an RCT. So approximately 200 patients were assessed and brought into the trial, and 164 patients were randomized, and all of those patients had a synovial biopsy prior to going into the different arms of the trial, where they were randomized to either rituximab or tocilizumab. Now, the primary aim of the trial was to determine if patients were B-cell poor, as determined by a histological marker. Then the hypothesis was that those patients would respond less well to rituximab, which is a B-cell depleting drug. And we hypothesized that they should respond better to tocilizumab, which targets the IL-6 pathway. As a secondary endpoint, we aimed to determine whether in patients who are B-cell rich, whether rituximab and tocilizumab would have the same effect. Now, it's important to note that the trial was not powered to determine if there was inferiority in the secondary endpoint. It was powered entirely on the B-cell poor group for looking for superiority of tocilizumab over rituximab. Now, when we analyzed the trial by histology, we could see that there were differences in terms of the response between rituximab and tocilizumab, but the trial did not meet its primary endpoint on the histological classification. However, built into the trial study protocol was a plan always to use RNA sequencing or a similar methodology to more accurately determine the level of B cells within the joint. And because this was a very novel trial, we went on to do this. And what we did when we analyzed the data from the RNA sequencing, we divided the cohort using the median so that the cohort division was unbiased. And using the heat map, you can see that on the left of the heat map, those patients with low levels of B-cell genes were classified as B-cell poor, whilst those patients with high levels of B-cell genes were classified as B-cell rich. And when the study was reanalyzed using this reclassification, we could see that the study met its primary endpoint, namely that in the B-cell poor group, 63% of the tocilizumab patients responded compared to only 36% of rituximab-treated patients. And this had a p-value of 0.035. Whereas on the right, if you look at the B-cell rich cohort, you can see that there was no statistical difference in the patients classified using this Phantom-5 B-cell module classification. So this is particularly informative because not only did it show that the initial hypothesis of the study was correct, but it suggests that RNA-seq classification appears to be superior to synovial histology for distinguishing these treatment endotypes. And this is some of the supplementary and secondary endpoints, and you can see the histopathology classification on the left and the molecular or RNA-seq-based classification on the right. And I think the most striking thing about this is that many more of those endpoints were met when using the RNA sequencing for defining the B-cell poor versus the B-cell rich. So just to sort of summarize, where are we now in terms of biomarkers in RA, and where would we like to be? Well, the key biomarkers that are in clinical practice that are widely used include anti-CCP antibodies and perhaps more recently some other antibodies such as carbamylated P antibodies. And also we have very good disease monitoring markers, including clinical metrics such as the DAS28 and CDAI, but we also have ultrasound and X-ray. However, in terms of actually stratifying patients, currently the best paper really is the Arthra paper with the B-cell gene module, and other methods have not yet demonstrated in anything like as rigorous a way as using a randomized control trial any difference in terms of stratification. But I think one key point to note is that in the end, we may have to rely on multi-omics and machine learning or artificial intelligence in order to actually develop good algorithms that can stratify patients to either methotrexate-based DMARDs or anti-TNF or other biologic markers. So just to summarize from this sort of section of the talk, overall, we could identify that there were pathotypes associated with RA, and these are linked to RNA-seq modules, and we've demonstrated that these are associated with different outcomes. We are able to improve upon that and develop panels using gene expression based on nanostring that could be used to stratify patients. We're also able to demonstrate in the Arthra trial that patients with low B cells in synovium were more likely to respond to tocilizumab than rituximab, which is perhaps the first demonstration of true stratified medicine in rheumatology. And we also demonstrated that RNA-seq classification was superior to synovial histology for distinguishing these endotypes. So overall, I would like to perhaps put it to you that biomarker-driven trials are going to be the future of rheumatology. And there's been huge amounts of interest from industry, from many different quarters. And I would say that this is really where the future of rheumatology trials is going. And already at the last ACR conference, we could see that patients were being stratified in randomized trials based on their interferon response. And so I think this really is going to be the future of rheumatology, is determining how different biomarkers can be used in clinical practice. And with that, I would just like to acknowledge all the different people and different groups that were involved in this work. Thank you.
Fantastic. Thanks so much, Dr. Lewis. So now let's move on to the Q&A. There's still time to submit your questions using the Q&A tab to the left of your screen. We have lots of great questions already, and we'll try to get to as many as possible. So our first question is, You've shown that autoantibody profiling stratifies SLE patients with a potential for differential treatment, and we have also seen patient stratification of Sjögren's patients using a similar approach. Do you see any utility in applying the autoantibody profiling approach to your RA patients to assess potential underlying predisposition to treatment options?
Yeah, this is a good question. I think it definitely could be applied, and we already know that anti-CCP antibodies are associated with worse prognosis and X-ray progression, and there is some evidence from trials that that patients who are treated with rituximab, who are CCP positive or rheumatoid factor positive, may respond better. Although I admit that there's some controversy over those results. But it wouldn't surprise me, given that some drugs target B cells and autoantibody secreting cells to a greater or lesser extent, if autoantibodies have an important role in terms of predicting who's going to respond to different treatments. So I suspect it would be very valuable to apply an autoantibody array to RA patients and see how they respond to different drugs. I mean, the only thing to note is that the autoantigens that may be informative may be different from the ones in lupus, since there are differences between the two diseases. But that's something that I think further research would be required to ascertain.
Great. Thanks so much. So our next question is, how would you describe the biological significance of using autoantibodies as biomarkers?
So the autoantibodies, it was interesting to see that there were differences in terms of functional pathways. We conjectured in the paper that because some of the autoantigens were associated with the TGF-beta and Wnt signaling pathway, that it may be that novel drugs that target those pathways might be more effective in that subgroup of lupus patients. I mean, in some respects, that may be a bit of an oversimplification, but it certainly raises that as an interesting possibility. In the same way that we're starting to see from the ARTHR-A trial that patients with higher levels of B cell are more likely to respond to a drug such as rituximab than if they had low levels of B cells. And so that finding already suggests that the level of gene expression within target tissues may be of relevance. And so it may be similar for autoantibodies. But I think this is something that we would very much like to do further work on. And the only way to determine that would be to actually measure those autoantibodies within clinical trials and then look at the response and actually determine if there was a difference in terms of response to different drugs relating to their autoantibody profiles.
Great. How do you envision patient stratification data being used in real-world situations?
It's a very good question. I think the whole aim of the precision medicine movement is to move towards some kind of either bedside or laboratory test that's relatively easy, so that patients can have a blood test. That would be ideal, rather than necessarily having some kind of tissue test like a biopsy, like a synovial biopsy. In an ideal world, the patient would have a blood test, and it might need to be some kind of mixed assay. Maybe it might measure some genes, it might measure some gene expression, and maybe it might measure some autoantibodies. But I suspect we will eventually move to a system whereby we're able to predict which drug patients are going to require. That's certainly what we'd like to see in the future.
Fantastic. So our following question is, Clustering of the novel autoantigens revealed 4 distinct SLE subgroups associated with 4 clusters of autoantigens. The identified autoantigens cluster into functional groups, hinting at differing underlying pathogen mechanisms and possibly at differing treatments for the SLE subgroups. Has any further work been done in terms of considering offering different treatment options according to the subgroups of SLE patients?
That's a very good question. Not yet, but it's something that that we would certainly very much like to do and be involved in. I will say that a number of different pharma companies have expressed interest in looking at the levels of autoantibodies in patients in clinical trials. So I think this is something that it's a matter of time, that as the autoantibody array gets tested in different clinical trial settings or in future prospective cohorts, where we can actually carefully measure response to individual drugs, that's when we'll find out whether we can predict response from the actual array.
Great. And our next question is, have the clinical outcomes of the SLE patients been assessed since the paper was published? It would be interesting to know if patients in the 4 identified subgroups have shown different clinical disease progression.
No, unfortunately, we don't have long-term clinical outcome data for some of these patients. Many of the patients that were assayed that were part of the UK cohorts largely gave a single sample because of being involved in genetic studies. And so unfortunately, we don't have long-term clinical outcome data on all of those patients. But it's something that we're looking to collect in the future, is to have a separate prospective cohort and actually perform antibody testing in that cohort.
Fantastic. So this will be our final question. We had a lot of great questions today and couldn't get to them all, but we will try our best to get back to everyone who submitted personally after the webinar. So our final question is, you have stratified RA patients for the rituximab study on their lymphoid versus myeloid cell infiltration into the synovium by sampling synovial fluid and RNA sequencing. Is it possible to use a less invasive procedure, perhaps a blood-borne marker such as CXCL13, to assess cell type? There is some literature that suggests that low circulating CXCL13 is associated with a better response to TNFA blockade.
That's a very good question there, and also topical. We have started to run assays for CXCL13 in some of our cohorts to see whether that can be used as a marker. The data suggests that soluble CXCL13 may be a marker for the presence of B cells within the joint, which is similar to saying that the lymphoid pathotype. So it's possible that CXCL13 might be used in place of doing, say, RNA sequencing or gene expression within the joint. But how accurate CXCL13 has yet to be determined. And I suspect that often when you use a single serum marker, it generally doesn't tend to be as accurate or as effective as if you use a panel of markers. So I suspect in the future we may need CXCL13 perhaps plus another couple of markers. So in the ADAPTER trial, they then went on to use CXCL13 and ICAM1, and that was informative. But I think other markers as well may help to improve how well that system can predict whether you've got B cells within the joint, and thereby whether you're going to respond to different drugs. For example, it wouldn't surprise me if a combined module that included CXCL13 and perhaps the interferon gene signature, for which there's already good evidence that the interferon gene signature can be used as a predictor of response to rituximab, for example, in RA. I mean, that may actually make a more accurate predictor. So I suspect a combined approach is probably the future.
Great. And with that, thank you for attending this Fierce Pharma webinar and submitting so many great questions. I'd like to thank our speakers for participating and Sengenics for presenting today's webinar. This webinar has been recorded. You will be able to access the recording within 24 hours using the same audience link that was sent to you earlier. Thank you again for joining, and we look forward to seeing you at future events.